In part one of this series on the innate immunity, I suggested that the human immune system is like a castle. A castle's main job was protection of a noble family and their territories from invading armies. I also suggested that castles have multiple lines of defense. The first line of defense consisted of elements that were built into the castle and provided barrier protection that had to be overcome during an invasion. The first line of defense included structures like curtain walls, gates, and gatehouses. Well, the body has barriers that must be penetrated by microorganisms. These include the skin and mucous membranes, and in the first part of this series, we actually talked about why the skin and the mucous membranes are so protective. Now, I also mentioned that if soldiers made it into a castle, there was a second line of defense that would jump into action. You had watchmen called sentries that would defend the castle in closer combat. In addition, you had archers that would rain down arrows on hordes that were close to breaching the castle walls. White blood cells, like neutrophils and macrophages, are part of the second line of defense in the body. They jump into action when microbes make it past the barriers, like the skin and the mucous membranes. In this video, we'll look at three other components that make up the second line of defense: inflammation, fever, and the complement system. By the end of this video, you should be able to describe the steps of an inflammation reaction and explain how it helps protect the body from infection. You should also be able to describe how fever and the complement system protect the body from infection as well. Inflammation. You've probably seen it on your body at some point during your lifetime. And when you see it, you might think to yourself, uh-oh, something is wrong there. Well, what you're really looking at is the body trying to heal and protect itself. Inflammation is characterized by 4 signs and symptoms. It's accompanied by redness, as you can see in this image, swelling, which is also called edema, pain, and of course heat. All of this is kind of like a side effect to what's really going on in your tissues. The whole point of inflammation is to destroy injurious agents or limit their effects on the body. What do I mean by injurious agents? Well, since this is a microbiology class, we're talking about things like bacteria, viruses, protozoa, fungi, basically your microbes. But keep in mind that inflammation may also occur when the skin is exposed to blunt force trauma, or it's exposed to toxins, or maybe it's been injured because you've had an unfortunate run-in with a knife while cooking, or maybe you've fallen and gotten an abrasion. Inflammation repairs and replaces tissue that is damaged by the injurious agent, whatever it happens to be. Now would be a good time to pause this video and go to the lecture videos folder in your course space. I've posted a video of me narrating an animation to describe what happens during an inflammatory response. Pause that video, then come back here and we'll review the steps of inflammation. So inflammation, again, is activated when proteins that are made by the liver and signaling molecules like your histamines, your kinins, your prostaglandins, leukotrienes, and cytokines cause vasodilation. So again, vasodilation is when your blood vessels, like capillaries, begin to increase in diameter. This also results in the separation of some of the endothelial cells that make up the walls of the capillaries. The result overall is that these blood vessels become leaky. Fluid from the blood, and in some cases blood, may begin to leak out of the blood vessels. And this is what gives us the red color that accompanies inflammation. It also is responsible for swelling. A side benefit of these leaky blood vessels is that phagocytic cells that may be in the area. These would be things like dendritic cells, to a lesser extent your mast cells, but also neutrophils and macrophages. They get recruited to that area. And these cells, remember, because you've been through phagocytosis by now, these cells have little receptors on them called pathogen recognition receptors, and they recognize PAMPs. These are pathogen-associated molecular patterns that are produced by the microbes at the site of injury. So the phagocytic cells will be drawn to that area because there's an increase in the number of molecules the closer they get to the area of damage, and they'll start to try to squeeze through the spaces between the endothelial cells and the blood vessels, and they'll actually enter the tissue. They will attach to the pathogens that are in the damaged area, and they'll begin to phagocytize them, which we've talked about. Margination is the term that is used for the phenomenon where these cells roll through the blood vessels, and chemotaxis is the term for their attraction to the PAMPs that are being released by the pathogens in this area. Diapedesis is the term for the squeezing of the phagocytic cells through the walls of the capillary and into the tissue. And then the next step, of course, is going to be phagocytosis. Now, what I haven't really talked about that also happens in this area around this time is that blood is clotting. Okay, so where there's damage to the skin, you will have blood clotting to kind of plug up the area of damage and to start to plug up the blood vessels as well. So the influx of blood and fluid is responsible for the swelling that we see during inflammation. It causes pain because remember there's nerve endings associated with the skin. So pressure on those nerve endings is interpreted as pain. And then you also have heat because again, blood is rushing to the area of damage. After the inflammatory response has removed offending microorganisms and damaged cells, repair can occur. Repair encompasses two different processes. You have regeneration. This is a type of healing in which new growth of epithelial cells restores portions of damaged tissue to normal state. So if you think about the last time you got a paper cut on your finger, and after about a week, it healed back to looking like your regular skin, that's regeneration. Replacement is another type of repair, but it occurs when you have severely damaged tissues or tissues that don't regenerate. And instead of getting a normal looking skin, you may wind up with a scar because the damaged area gets infiltrated by fibroblasts, which make a number of protein fibers, and they try to reinforce the damaged tissue. And that scar may eventually be replaced, but more often than not, it doesn't get replaced. Fever is another component of our second line of defense. It's described as an abnormally high body temperature. You'll probably remember from A&P that the temperature of the human body is usually within 1 degree of 37 degrees Celsius. This temperature is maintained through homeostasis and is controlled by the hypothalamus portion of our brains. When there's an infection in the body, chemicals called cytokines are released by our white blood cells. You can think of cytokines as being communication molecules used by the immune system. A set of these are called pyrogens. Let me see if I can write this. Pyrogens. Pyrogens trigger fever in the human body. One pyrogen is called interleukin-1, or IL-1 for short. When IL-1 is released into the blood, it travels to the brain and it resets the body temperature at the hypothalamus. The hypothalamus then sends impulses to our blood vessels to cause them to constrict. And it also sends impulses to our skeletal muscles to cause shivering. Together, vasoconstriction and shivering raises the body temperature and results in chills. Fever can support the immune system's attempt to gain an advantage over infectious agents like bacteria and viruses because it makes the body less favorable as a host for replication. That elevated temperature causes viral and bacterial proteins to denature easily, which inhibits growth, and it gives the immune system a chance to jump into action and fight the infection. After the infection is over and it's been cleared, body temperature begins to fall. This is called a crisis, and vasodilation and sweating occurs to help cool the body. The complement system. This is so cool. So the complement system is part of your innate immunity. It's a second line of defense system. Basically, it's a set of proteins that are produced by the liver, and they assist the immune system in destroying microbes. They act in a cascade, so that means that there's going to be one chemical reaction which triggers a second chemical reaction, which triggers a third chemical reaction, which triggers a fourth chemical reaction, and so on and so forth. And this process is called complement activation. So there's 3 ways that the complement system can kill or help in an immune response. One way, and this is my favorite way, is called cytolysis. Here you have a bunch of complement proteins, and you'll know these when you see them because they're always designated with an uppercase C, and then they have some number after it. The number comes from the order in which they were discovered. But you start out with complement proteins, and they basically assemble in a prescribed way to form a pore or a channel in the surface of a bacterial cell. Enough of these form, causing the bacterium basically to explode. So cyto means cell and lysis means to break open. That's exactly what the complement system does here. It's going to break open bacterial cells. Another way that the complement system helps in treating an infection, it helps the immune system to overcome an infection, is through something called opsonization. So remember, opsonization is a process whereby proteins or molecules called opsons attach to the surface of a microorganism and make it more visible to the immune system. The opsons essentially mark the cell for destruction. So here you have complement proteins that bind to PAMPs on the surface of a microorganism, and it makes that microorganism more easily recognized by phagocytic cells, and then the phagocyte basically engulfs the microbe and destroys it. The third way that the complement system can help in an infection is that it can stimulate inflammation. So you have complement proteins that will activate mast cells. Mast cells produce histamine, and then the histamine triggers inflammation. And as we've already seen, inflammation allows phagocytic cells like macrophages and neutrophils more access to body tissues, and they phagocytize invading microbes. Finally, I want to remind you of interferons. We talked about interferon alpha and beta already. These are antiviral molecules that are produced by infected cells. Remember, during a viral infection, a virus actually enters a host cell and it begins its replication cycle. During this process, transcription of alpha and beta interferons occurs. The infected cell secretes these alpha and beta interferons into the environment around it, and they bind receptors on adjacent cells that have not been infected. The binding of the alpha and the beta interferons stimulates these cells to produce antiviral proteins that can interrupt the virus life cycle if the second cell should become exposed to a virus. So you can also consider these interferons as being antiviral, of course, but they're also part of the second line of defense. I don't know if you remember, but there's also, this is interferon alpha and beta, but there's a third interferon called interferon gamma. And what it does is it causes neutrophils and macrophages to kill bacteria. So it's also part of the second line of defense. So now, between the first part of this video series and the second part, I hope you have a little bit of an idea of how your body is able to protect you against infection by microorganisms. We've talked about the innate immune system here. Remember, the important thing about the innate immune system is it's the same for everybody. It basically fights infection the same way. It recognizes groups of microorganisms, such as bacteria, or viruses or protozoa or fungi, and then it treats all microbes, or it fights rather, against all microbes that are of that type in the same way. It's not specific to what kind of microbe is causing an infection. In this video, I've talked a little bit about how inflammation helps fight infection, primarily by increasing blood flow to a site of infection or tissue injury, and then allowing macrophages and neutrophils and other phagocytic cells to gain access to that area of damage where they can fight any microbes where they can phagocytize any microbes that might be present, and they can aid in tissue repair by removing dead and damaged cells. I've also talked a little bit about how fever and the complement system protect the body. Remember, fever raises body temperature, making the human body an unsuitable environment for growth of bacteria and viruses. This gives the immune system a chance to find the microbes and phagocytize them, kill them. And then you have the complement system, which is basically a set of proteins that are made by the liver, and pretty much everyone's liver makes these. And they have three different ways that they fight infection. One, you have the cytolysis, where complement proteins assemble onto a bacterial cell, punch holes in its surface, causing it to explode or lyse. You have opsonization, where complement proteins attach to the antigens or PAMPs on the surface of a bacterium and make that bacterium more visible for phagocytic cells. And then that leads to phagocytosis. And then you've got the final way it can trigger inflammation, which we've already talked about. So I hope you have a better idea of how the innate immunity protects you against infection. You will need to be able to talk about the steps of inflammation and why they're important. You'll also need to be able to describe fever, the complement system, and you could see a question about interferons as well on your final exam. All right, if you have any questions, don't hesitate to reach out. In the next video, we're going to look at the adaptive immune system. I'll see you there.